In the following slide, I will explain the difference in the protein translation of prokaryotes and eukaryotes. Firstly, in prokaryotes, the shine-Delgano is a ribosomal binding site that is only found in the messenger RNA of prokaryotes. It helps start initiation of protein translation, as this is where the 30S ribosomal subunit aligns itself. The shine-Delgano is usually located just upstream of the AUG initiator codon and in between two non-coding regions. In comparison to the shine-Delgano site, eukaryotes have a 5' cap at the end of the mRNA. This is a 7-methyl guanosine structure, which is important for many reasons. These include translation initiation, as the 40S ribosomal subunit binds to this cap. It also acts as protection for the 5' end of the mRNA. Eukaryotes also possess a 3' poly A tail, which prokaryotes do not have. This is added during transcription and is made up of a series of adenine nucleotides. These add stability to the mRNA, aid in translation termination, and act as protection for the mRNA like the 5' cap. The length of a poly A tail is usually approximately 200 bases, but this length decreases with age. As I said, the 30S small ribosomal subunit is the first to attach to the mRNA. This is then joined by the 50S larger subunit. These work together and form a 70S ribosome which moves along the mRNA to synthesize proteins. These are made up of both ribosomal RNA and proteins, which make up the 70S. Like in prokaryotes, the smaller ribosomal subunit attaches to the mRNA first, but in eukaryotes this is a 40S subunit. It is then joined by a 60S larger subunit. They move together along the mRNA as an 80S unit. Another major difference is the place where the protein synthesis occurs. In prokaryotes, both transcription and translation occur simultaneously in the cytoplasm. However, in eukaryotes, transcription takes place in the nucleus. The mRNA then travels to the cytoplasm where protein translation takes place. Protein synthesis inhibitors can stop translation at many stages, including at initiation stage, when tRNA is binding, when translocation is occurring, and at the peptide bond formation stage. These inhibitors can include streptomycin, tetracycline, chloroamphenicol, and diphtheria.